Moderator on for this session. Obviously, I'm not Jeff. I'm not nearly as good looking or smooth as he is, so bear with me. I also wasn't prepared to do this session, so I had to dress down a little today. There was a dude wearing a hoodie yesterday, so I figured the bar was kind of low, so I'm a little bit above that. We're all right. Okay. Our first presenter is Michaela Dustin. She recently graduated from the University of Auckland with a postgraduate diploma in forensic science in 2014. She obtained her undergraduate degree in chemistry and biochemistry from Massey University in 2013. She joined ESR, the Institute of Environmental Science and Research in Auckland, New Zealand, as a senior technician on the physical evidence team in July 2015. In this role, Ms. Dustin provides technical support to senior scientists, predominantly on trace evidence analysis, such as hydrocarbon fuel residues, condom lubricants, defense gas sprays, paint, and glass. Her presentation is titled Further Persistent Studies on PDMS Condom Lubricants. Please welcome Michaela Dustin.
Cool, so thank you. Good morning, everyone. Apologies, I've got a bit of a head cold, so hopefully I go all right. So as you can see, I'm going to be talking about the persistent studies of PDMS condom lubricants that have been carried out in my laboratory over the last few years. So I work at the Institute of Environmental Science and Research Limited, known as ESR. ESR is one of 7 Crown research institutes in New Zealand, and we specialise in science relating to people and communities. ESR consists of multiple groups including health, food, water and forensic science. Our forensic science group is then split up into multiple teams, including the physical evidence team, which I'm part of. And one of the main evidence types that we're currently asked to analyse for is condom lubricants. And there's New Zealand down there, if you don't know where we are, for Australia. So analysis of intimate swabs from alleged sexual assaults for the presence of condom lubricants is becoming a routine request for many trace forensic laboratories. The increasing public awareness of the high evidential value of DNA has seen an increase in the use of condoms by sexual offenders. Analysis for condom lubricants can provide scientific evidence to collaborate or refute a given scenario. Persistent studies of such lubricants are vital to support the interpretation of forensic samples analysed for the presence of condom lubricants. So the majority, more than 95% of condoms in New Zealand are lubricated with polydimethylsiloxane, known as PDMS, with fewer containing water-soluble lubricants such as polyethylene glycol, known as PEG. So intimate swabs are routinely analysed for the presence of PDMS at ESR by extracting the swabs with hexane and analysing these extracts using pyrolysis gas chromatography mass spectrometry, known as PGCMS. So pyrolysis of PDMS converts the polymer into cyclic oligomers. And this series of dimethylsiloxanes of increasing molecular weight can be separated and detected by GCMS. The smallest cyclic oligomer detected is hexamethyl cyclotrisiloxane, or D3 for short, with the oligomer series usually ranging from D3 to D7, where D7 is tetradecamethyl cycloheptasiloxane. This figure on the left shows the PDMS molecular depolymerization mechanism. And at ESR, to identify PDMS in a sample, the D3 cyclic oligomer and at least 2 other cyclic oligomers in the D4 to D7 range must be present. Confirmation of each compound requires the presence of the molecular ion isotopic cluster and at least 3 additional abundant ions in the mass spectra. In this figure on the right, it's not very clear on the screen, but it shows the pyrogram of a standard solution of PDMS in hexane with the cyclic oligomers identified. To date, the majority of research relating to the forensic analysis of condom and sexual lubricants has focused on evaluating and validating different analytical methods. Very little research has been published relating to the transfer and persistence of such lubricants during a sexual assault. In addition, most of the previous research has focused on the persistence of PDMS in the vagina. This presentation will include the results of research focused on investigating the persistence of PDMS on the penis and the mouth and on skin. We've also sampled a range of personal care products to get an idea of how widely spread PDMS is in common personal care products that are used on the body, and whether or not we can discriminate condom PDMS from other sources of PDMS. This research also investigated whether PDMS is present on tampon products available on the New Zealand market, and whether PDMS can be detected on intimate swabs that have previously undergone DNA extraction. So this work was made possible by students that have worked for us over the years, which included a master's student and a number of intern students. So most of these students were with us for less than 6 months, and finding volunteers to take part in these studies has been a bit of a struggle. Therefore, we've had limited time and limited sample numbers. However, from the small number of samples we have managed to obtain, we have unveiled a number of interesting findings, and this has helped us in the interpretation of case results. For our study of persistence of PDMS on the penis, we had 6 volunteers who applied a condom to their penis for a few minutes. After they removed the condom, they self-swabbed their penis at specified time intervals and recorded their activities during this time. So we received a total of 14 swabs covering time intervals from 0 to 20 hours, and we found that PDMS was detected for up to 20 hours, the longest time interval studied, with some variation between participants. This included PDMS being able to be detected after activities such as sleeping, urinating, washing, and exercising. For our study of persistence of PDMS on the skin, we chose to transfer PDMS to the inside of the forearm. This area was selected as being a relatively hair-free, easily accessible region of the body. Previous case samples have included swabs taken from the inner upper thighs of complainants, and it was thought that the inner forearm would be suitable as a substitute for this area. The skin of the volunteers' forearms were marked using a marker pen into areas of approximately the same size. Prior to the transfer of PDMS, a control sample was taken by swabbing the inner forearm with a wet swab. And wet swabs were used as preliminary trials determined that wet swabbing recovered approximately 3 times more PDMS from skin than dry swabbing. A condom was then placed on a carrot or cucumber and firmly swiped against the volunteer's inner forearm. One area of the forearm was swabbed immediately for the initial trials. Volunteers then self-swabbed each marked area of their inner forearm after specified time intervals. The volunteers were asked not to wash themselves or use any personal care products that contained PDMS or other silicone-containing compounds during the time intervals. For the initial study, we had 10 volunteers covering time intervals from 0 to 12 hours, and we found that PDMS was detected up to 12 hours, the longest time interval studied. So given these results, we wanted to do a further study and target longer time intervals. For these trials, we had another 10 volunteers covering time intervals up to approximately 48 hours. Due to these long time intervals, it was seen as unrealistic to ask volunteers not to wash during these time intervals. But once again, the volunteers were asked not to use any PDMS containing personal care products. So we found that PDMS was detected up to 52 hours, the maximum time interval studied. The maximum detection time observed for the retention of PDMS varied between volunteers from 24 to 52 hours. However, intrapersonal variation was also observed. For example, for one participant, PDMS was detected at 48 hours, but swabs taken at 35 and 36 hours were negative for PDMS. And also of note was that PDMS was detected on a number of control swabs taken from volunteers prior to PDMS being transferred to their forearms, suggesting that these people may have got PDMS on their skin from a personal care product. In these instances, the subsequent persistence trial swabs were not analysed. So these results indicate that PDMS can be retained on the skin for up to 2 days. For casework samples, especially when there is a significant time delay between the alleged incident and the swabs being taken, efforts should be made to establish whether or not the complainant has used any PDMS containing personal care products during this time. For our study of persistence of PDMS in the mouth, a blank swab was taken from the mouth of each volunteer by asking the volunteer to rub a cotton swab against the inside of their cheeks and on their gums. A condom was then placed on a carrot, and the volunteer moved this around in their mouth for about a minute to simulate oral sex. They then self-swabbed the insides of their cheeks and gums after specified time intervals. For the initial study, the volunteers were asked to abstain from eating and drinking during these time intervals. For the 5 volunteers that provided samples for this persistent study, we found that PDMS was detected after 2 and 4 hours. And when the PDMS transfer occurred directly prior to the volunteer sleeping, PDMS was detected on the swab taken directly after waking at 8 and 9 hours. From our next study on the effect that eating and drinking has on the persistence of PDMS in the mouth, we found that PDMS was not detected after the participant ate or drank within 1 hour of PDMS transfer. This finding suggests that the action of eating or drinking removes sufficient PDMS from the mouth to an extent that it can no longer be detected. This finding is useful for the interpretation of casework samples in that PDMS can be detected on oral swabs taken from complainants if the medical examination is performed within a short time frame of the alleged incident and if the complainant has not consumed any food or drink. So for our study of personal care products for PDMS, 178 products were analysed, such as shampoo and conditioners, moisturisers, hairspray, shaving foam and sunscreen. PDMS was detected in 98 of these products, so approximately 55% of the products that we analysed, which gives an indication as to how widely spread PDMS is in common products. Other compounds were often present in the output chromatogram, which would enable us to discriminate these as non-condom PDMS, assuming that these additional compounds are not preferentially absorbed by the body. So the issue of discriminating PDMS from a source of condom lubricant compared to another personal care product remains an ongoing issue, with further research needed regarding what, if any, compounds are differentially absorbed by the body. So we then wanted to survey some tampons to see if they could be a potential source of PDMS. So a range of tampon products on the New Zealand market were donated by colleagues. 1 tampon per product was analysed. We ended up having 11 different tampon products that were analysed, and with a couple of the tampons came an applicator, so we also analysed these applicators as well. No PDMS was found on any of the New Zealand tampons or the applicators that we analysed. This finding is significant as it demonstrates that tampons are not a potential source for PDMS. So we occasionally get swab samples to test for PDMS after they have come from the biology team, where they have been extracted and analysed for DNA. We previously have been analysing the swab head remains as per usual, and also analysing the supernatant from the DNA extraction process. We were confident that we could detect PDMS on the swab remains if it was there originally, as the aqueous DNA extraction process was unlikely to remove PDMS. Previously, we also tested the supernatant just in case any PDMS had been removed from the swab into the extraction buffer during the DNA extraction process. So a number of swabs were loaded with known volumes of a PDMS solution. 1 set of these swabs were sent off to biology for their standard DNA extraction process, and the other set were retained as the control swabs. So the remaining swab heads, the supernatant from the DNA extraction process, and the control swabs were all analysed for the presence of PDMS. PDMS was found on the swab remains that had previously been extracted for DNA at similar levels to the respective control swabs. PDMS was not detected in the supernatant samples that came from the swabs loaded with low to medium amounts of PDMS, and low amounts of PDMS were found in the supernatant samples from the swabs loaded with high amounts of PDMS. So these results indicate that intimate swabs could be extracted for the presence of DNA prior to the swab remains being analysed for the presence of PDMS without any significant loss of evidence. It also suggests that there is little to be gained from also extracting the aqueous supernatant, since the majority of PDMS remains on the swab remains. Therefore, we have changed our protocol and now just analyse the swab head remains and do not worry about analysing the supernatant. So in conclusion, the key findings we found were that PDMS can persist for up to 20 hours on the penis after a condom has been worn and can still be detected after activities such as showering and exercise. No longer time intervals were studied, and therefore it may be that PDMS can persist on the penis for even longer periods of time. And this finding would suggest that it is worthwhile swabbing the penis of a suspect if condom use is alleged by either party. PDMS was shown to persist on the skin of the inner forearm for up to 24 to 52 hours, with variations seen between the different volunteers. PDMS on the skin is not significantly affected by soap and water, and background levels of PDMS were found on some volunteers, which shows the importance of having controlled skin swabs or finding out whether the complainant has used any personal care products that could contain PDMS. PDMS transferred to the mouth was shown to persist for at least 4 hours if the volunteer did not eat or drink, and at least 9 hours if the volunteer slept. However, PDMS was unable to be detected in all trials after the volunteer ate or drank, indicating that PDMS in the mouth is significantly affected by eating and drinking. Therefore, while it is recommended that oral swabs be collected from complainants in cases where oral sex with a condom is alleged, it is likely that PDMS will only be detected if the medical examination is performed within a short time of the alleged assault. Interestingly, of the 2 cases submitted to our laboratory that included oral swabs, PDMS was detected on one swab collected 2 hours after the alleged incident, and PDMS was not detected on the other swab that was collected 7 hours after the alleged incident. So the aqueous extraction of intimate swabs for DNA analysis prior to analysing the swab remains for PDMS was found to not be detrimental to the detection of PDMS. This finding means that one swab can be analysed for both DNA and PDMS. However, any water-soluble lubricants such as PEG or glycerol would be affected by the aqueous DNA extraction method. Transfer and persistent studies are essential for the correct interpretation of any type of trace evidence. The results of these studies add to the growing body of knowledge on the persistence of PDMS on and in the human body. However, we would encourage other laboratories to carry out similar studies to ensure that a wide range of scenarios are covered. For example, there is a lack of research looking at the persistence of PDMS in the anus and rectum, and the number of vaginal persistence studies is relatively small. We would also like to extend our number of samples pertaining to background levels of PDMS on random man or woman, in both bodily cavities and on the skin. So I'd like to thank all the people that made this work possible, including all the students that we've had over the years to coordinate and analyse all the samples, all the volunteers that provided us with samples, physical evidence staff for supervising students and interns, Forensic Technology Centre of Excellence for hosting this event, and the National Institute of Justice and RTI International for funding my attendance. Thank you.